offline. Thank you.
Thank you very much.
The last talk is from local, from Philippe Menasche. He's going to talk about pluripotent stem cell in clinics.
Where's the pointer? Thank you very much. Well, first of all, thank you to Dr. Song, GQ, and Barry, obviously, for the pleasure, the privilege of this invitation. I really enjoy now starting collaborating further with this group. So as you can guess from the title, which was allocated to me, there will be two parts. First, I will briefly summarize the results of the embryonic stem cell trial, because much of this material has already been presented, and then I'll like to have a slight glimpse into the future, talking about what we're currently doing. So briefly, to remind you the protocol of this clinical trial, we started from human pluripotent embryonic stem cells, scaled them up, then induced cardiac differentiation by a 4-day exposure to 2 cytokines, primarily BMP2, ended up with these unsorted cardiovascular progenitors, which were then purified by immunomagnetic technique using SSAA1 as a marker for the loss of pluripotency. And when we looked at the transcriptome of these progenitors, it turned out that, as you can see, the pluripotency genes, which were obviously present in the starting material, had been knocked down in the population of the cardiovascular progenitors, and conversely, these cells did express the mesodermal cardiac markers, which were not present initially. Well, then we mixed this progenitor cells with fibrinogen and thrombin, so as to make a patch, a fibrin patch, which was then delivered during a conventional CABG procedure. And these cells were carefully screened for the absence of contamination and, still more importantly, for their cytogenetic stability. And that was done by a combination of techniques, karyotype, FISH, focusing on chromosomes known to be unstable during culture, and finally, CGH arrays to ensure that really the cells had remained cytogenetically stable. So then we proceeded to the clinical trial. That was a first-in-man safety pilot trial, just including 6 patients requiring CABG and with severe left ventricular dysfunction. And this is just a picture of the operation. This is not a cell sheet, but it looks like it's a fibrin patch with the cardiovascular progenitors embedded in the patch, which was then delivered onto the epicardium of the infarct area. And then I took a piece of the patient's pericardium and used it as a flap to cover the fibrin patch for 2 reasons. Number 1, for securing the fibrin patch in place, and number 2, for hopefully providing the cellular graft with the trophic factors which are present in the pericardium. So the first important result was related to the feasibility. Clearly, it is feasible. You can end up with a highly purified population of progenitors. You see over 90% SSAA1 positive cells, which, as shown by the PCR, which were done systematically before each lot release, show the downregulation of NANOG, taken as a marker for pluripotency, and conversely, the overexpression of islet 1. Now, obviously, that was a safety trial, and so we were concerned about 3 major potential complications. Number 1 was arrhythmia, and that's the reason why all patients were fitted with an internal defibrillator. And when we looked at the serial interrogations of the device, so far and so good. There's been no episode of arrhythmia in any patient. The last patient is supposed to come back next week for his 1-year follow-up, but as far as we know, is doing well. Second safety issue, obviously, is the tumor, and that's why these patients underwent fairly extensive imaging studies before surgery, after 6 months, after 1 year, and again, so far, we haven't seen any uncontrolled proliferation either in the heart or remotely. The third potential concern was alloimmunization. What you see here is that actually these cells do express HLA-1. They do not express HLA-2 or co-stimulatory molecules. And interestingly, when you stimulate them with interferon gamma, they overexpress HLA-1, which might be a good thing in terms of protection against NK-mediated injury. When we did MLR in these patients, we observed, as you can see, that actually the responses were weak, supporting the low immunogenicity of this patient population. Only 2 patients had a slightly higher response, but much lower than the one you get in the control patients, well, the control samples, that is when you mix mononuclear cells from one patient to another. And actually, 3 patients over the course of the trial developed alloantibodies against specific antigens expressed by the cell line. But I draw your attention to the fact that the higher value MFI, mean fluorescence intensity value we got was 4,000, which is still below the threshold which has been reported to be cytotoxic. So overall, no real issue, and all these episodes were clinically silent, no issue related to alloimmunization. As an additional safety measure, we try to see whether some cells had escaped from the patch and were circulating in the patient's blood. So we screened the patient's blood by sequencing, and we were lucky enough because it turned out that the cell line had polymorphisms which are fairly uncommon. And as you can see, none of our patients had such a polymorphism before the operation, and we equally failed to detect one after, suggesting that migration of the cells was quite unlikely. And this is just one example. You see here the ratio of the sequence between C and T, and you see how different it is in this particular patient. So it's unlikely, once again, that cells had migrated. Well, efficacy was not an endpoint. I just show you the increase in ejection fraction, which for me is completely meaningless since these patients also got coronary artery bypass. More interesting maybe is the changes in the regional wall motion, focusing on the segments which have been cell and patch treated, in particular because most of these segments had not been revascularized, but I think that one should be extremely cautious in the interpretation of these data. Once again, the study was not designed to show any evidence for efficacy. And so to conclude, I think the best statement which can be made is the one of the editorial which was published at the time this paper recently appeared in the journal, in the JACC. And I don't think that so far we can go further except to say, yes, it is feasible, yes, it looks safe. Now it remains to be really demonstrated whether it's efficacious. So now, how does it work? Well, one possibility, obviously, is that the cells that we injected made a new myocardium. We know that these cells are able to contract. These are the cells that we actually transplanted if you let them go until the stage of the beating cardiomyocytes. I think, unfortunately, that this is wishful thinking, and that it's more likely, as repeatedly said throughout this symposium, that the primary mechanism of action is paracrine through the release of factors activating endogenous reaper pathways. And we have heard several presentations showing that probably many of these factors are clustered in extracellular vesicles, not only exosomes, but also microparticles. We know that these particles are able to deliver their cargo in the recipient cells. Well, what I would like to share with you now is a recent video microscopy from the lab, just documenting the reality of this internalization of the vesicles into the recipient cells. So these are the vesicles which have been labeled in green, and if you follow the arrows, you will see that, and this is real-time assessment, that they finally get into the cells where they are supposed to deliver their payload. Now, what is the payload made of? MicroRNAs, obviously, proteins, lipids, and so on. These factors are going to activate endogenous pathways. We don't know exactly which ones, but one has been of interest for us, and this is the possibility of the generation of new cardiac cells, but new cardiac cells coming from the host, endogenous sources. And to try to have a look at that, we use this fate mapping Krelox model, where if you give tamoxifen, the existing cardiomyocytes will turn in green. Well, this is a myocardial infarction model, and you see that after a controlled injection in the area of infarction, not unexpectedly, you do not have cardiomyocytes. Now, what happens now if you inject cardiovascular-derived extracellular vesicles? Well, you see here that in the infarct area, you have troponin T positive cells, so likely cardiomyocytes, but they are GFP negative. GFP negative suggesting that they were not pre-existing to the injury and that they may come from endogenous sources that we're currently trying to characterize. Conversely, here in the peri-infarct area, you have also cardiomyocytes, but they're GFP positive. So to try to assess a little bit further the effects of these vesicles, we first did the study in which we took the cardiovascular progenitors, the SSA1, exactly those that I had transplanted in the patients. We cultured them for 48 hours. Then we collected the medium, isolated the vesicles, and at the same time, obviously, collected the cells themselves. And then we designed this very simple mouse study in which the treatment was delivered 3 weeks after the infarct. I think it's important because this is really the setting of the chronic heart failure, different from acute MI. And as you know, in the mouse, if you do the infarct and then go back with surgery, mortality is pretty high. So to avoid that, we use the percutaneous echo-guided injections with virtually no mortality. And so that was a head-to-head, really head-to-head comparison of the cardiovascular progenitors or the vesicles. And that was the same batch, and I focus on that. Same batch of cells, half being transplanted, half being used for collecting the medium and isolating the vesicles. And that was a randomized double-blind study. We took left ventricular and systolic volume as the primary endpoint, which I think is more reliable than ejection fraction. And this is the result. The functional outcome was absolutely the same with the cells and the vesicles. And when we looked at the signaling pathways that had been turned on in the recipient hearts, we found that basically similar pathways had been activated. So next we proceeded to another confirmatory study in which we simply changed from embryonic stem cells to iPS. We scaled them up. This is part of collaboration with Cellular Dynamics International, which provided these cells. They were differentiated, again, in cardiovascular progenitors, you see here. And this is how the exosomes look like. Then we characterized the cargo, and in particular the content in microRNA, found 16 overexpressed microRNA, and it turned out that the predicted targets went into the direction of an improved cardioprotection. I just draw your attention to the fact that one of the microRNA was 103. You will understand why in a few seconds. Then we did bioassays to assess the effects of these vesicles, and for that, used this conventional method in which you culture rat cardiomyocytes. They proliferate initially. It's a line, an immortalized line. Then you deprive them from serum, so you decrease the viability, and we expose these cells to increasing concentrations of vesicles, and you can appreciate the dose-dependent improvement in cell viability. Well, interestingly, it turned out that the 103 microRNA has among its target one protein which reduces apoptosis, and that suggests this kind of mechanistic link between the microRNA content and the target effects. Then we delivered these vesicles in vivo, and again, that was the mouse model, and what you can see is that we got quite nice results with the vesicles. And interestingly, again taking ventricular and systolic volume as a primary endpoint, you will see that the data are much more homogeneous with the vesicle compared with the cells, the progenitors. Here the data are more scattered, and this makes sense because cells tend to clump into the syringe. It's more difficult to have reproducible injections, whereas with vesicles you have a liquid, and so the injection is more straightforward. And finally, we looked at the transcriptomic changes in the target cells, and to make a long story short, found again that the pathways that had been turned on in the vesicle-treated group went into the direction of an improved cardioprotection. So these are the potential advantages of these vesicles over cells. It's clear, as mentioned by Gordana, that there are still a lot of translational issues, but it's interesting that this data fit with Gordana showed this morning and Professor Young yesterday. And so this is the reason why we're now in the process of translating this data to set up a phase 1 clinical trial. Thank you very much.